So we had previously approached week 3 by looking at all of the figures together. So the figures from, let's take a look at the name files. The figures from week 3 come from these two PDFs. And the first one is endocrine pancreas and then insulin action. The purple blocks, the purple borders here are for endocrine pancreas, so this first file, and then the ones in orange were the ones for uh, insulin action. So step 1, we'll do our medical terminology word of the day. Same thing. Oh yeah, yesterday was respirovirus, today is retrovirology. So yeah, moving on then, I found a better way to arrange the figures to make them flow better. So here's figure 1, which is the structure of insulin, and it's essentially the stages it goes through for processing. Figure 2 was the structural components of the pancreatic cell. Figure 3 is glucagon, and again, the stages it goes through for synthesis. Figure 4, what is this? The control of insulin synthesis and secretion. And number 5, this one was kind of not too complicated, and it was kind of integrated into the other one, but that's it for that. And then for the next PDF file, insulin action, remember figure 1 was this, a very zoomed in view of the receptor, which they eventually it ends up being called IRTK. What does it stand for again? IRTK. IRTK stands for insulin receptor tyrosine kinase, IRTK. And then there's a left and a right, cool. And then figure 3 is again an emphasis on the IRTK, on the let's call it ERTK. Yeah, there's an emphasis on the IRTK in the ERTK, and the emphasis, or what's being highlighted here, is insulin action and how it performs an import of glucose through this transporter here. Finally, figure 5 was this mechanism by which insulin promotes cell growth. And at the end, there were a lot of pages which did not have any figures associated with them. They were tables, but they for that last portion, those several last pages, I just made this separate section because there were no figures associated with it. So from the top, let's go back up to the top, right here. So when it comes to the structure of insulin and its synthesis, it's important to note the following summary. So there's some functions right there, function of insulin. Function number 1 is the promotion of anabolistic processes, so building stuff up. When it comes to the second function, there's inhibition of catabolism processes. So you promote the buildup and you inhibit breakdown. And something that will be relevant later when it comes to GCAG, this is my shorthand notation for glucagon. It's everything is essentially reverse in terms of the effects, so we'll get around to that. That will be very relevant later. So yeah, all of this is still relevant to figure 1. There's more details about insulin. The insulin metabolism. Okay, so there's a third function. It promotes the storage of fuel. Function number 4 is that it promotes the uptake of A acid, or aka amino acids, on the cells. And also function number 5, there's another promotion of uptake, but in this case it's the uptake of glucose. Moving on, when it comes to the production of insulin, it happens at the pancreas cell. So let's add the little symbol at the pancreas cells. Yep. And there's pre-proinsulin. Let's highlight that. There's pre-proinsulin, and then there's proinsulin, and then there's just um, regular insulin. This is the mature insulin. And some stuff to this stuff becomes relevant later. So we'll come back. We'll actually circle back to this because this is stuff that came later in the section, but it fit best here in this area that talked about the production of pancreas beta cells. So moving on to figure 2. Here we discuss the structural components. Okay, so this is a single beta cell drawn out schematically. So remember, you have the production of insulin at the pancreas beta cells, and I have color coded the various stages with these colors: red, or yellow, green. So step 1 is CRIP, aka transcription. That's pretty straightforward. And then lati, as in translation. This happens at the rough ER. So again, we'll draw the at symbol, rough ER at symbol. Cool. Step 3, and all of this correlates to the steps highlighted here. So step 1, we said CRIP for transcription. Step 2, lati, translation. Number 3 is just being transported from the ER to the Golgi. There you go, going from the ER to the Golgi. And then step 4 is secretory granules, the synthesis. You synthesize secretory granules. And then step 5 right here, the secretory granules experience maturation. And during step 6, you go from the something to the plasma membrane, I guess from the Golgi. So I'll fill that in later as these notes get fine-tuned. And then step 7 is exocytosis, when the proper signaling, when the proper signals are received. So there you go. That's figure 2. Cool. Some other notes related to that are as follows. If a patient does not have an insulinoma and they perform an overnight fast, there will be low amounts of C-peptide. However, if a patient does have an insulinoma and they perform an overnight fast, there will be high amounts of C-peptide. So when there is a reduction of beta cell mass, it means that there is not a production of insulin, and that's the definition of diabetes 1. So this reduction happens because of genetic predisposition, and there is autoimmunity related to that. Let's see what else is happening. Autoimmunity. So then we can get into details about genetics. So with diabetes 1, there is a gene named HLA. I'm pretty sure from the immunology class that stands for HLA. It stands for human leukocyte antigen. So there's two scenarios to consider whenever the HLA gene is involved and whenever it is not, when it's not involved. So moving on, now here we dive into diabetes 1, diab 1 details. So you have insulin. They require insulin, which is exogenous, because I remember diabetes 1 because I focus on the number 1, and it's the problems arise because there is one organ which is not performing its job appropriately, and that's the pancreas. And as we mentioned here, there was autoimmunity, and that's what destroys the pancreas. So there is one organ so involved with diabetes 1, type 1, and then diab 2. 2 is plural, so it's, I think, of the plural number of cells throughout the body. It's just, a numerous amount which experience insulin resistance. So the number is not one, so it's not just localized to one place at one organ. Now we're dealing with a variety of cells, multitude of cells. So that's how I remember diabetes 1 versus 2. So next you have next here we have notes about normal, the normal insulin. Your normal, we can call it normal slash natural, because here in this lecture we there's a table. We learn a lot about insulin, which is synthetic, or other drugs which can mimic the action of insulin. So in this case, we're talking about natural insulin or normal insulin. So it's classified as fast acting, and it's used to treat these three conditions. So there are insulin analogs which fall under one out of three categories. So you can also have fast, and then medium, or that's what I call it. They call it intermediate, and then long. I You could also, I think of this as slow, long acting or slow acting. Cool. So here are some details about diabetes 2. This is just a direct screenshot from the PDF because there were not that many details related to this. So I think it was a quick overview, and it was just it seemed more worthwhile to simply copy this. So let's read this verbatim. Type 2 diabetes mellitus occurs when peripheral insulin resistance leads to relative insulin deficiency. In a patient with type 2 diabetes mellitus, the C-peptide may be elevated at the outset of the disease when the patient is hyperinsulinemic due to hyperglycemia. However, over time, insulin secretion, and hence the secretion of C-peptide, often diminishes because the pancreatic B cells can become overtaxed and respond by decreasing their amount of insulin. Hence, measurements of C-peptide may help to monitor the progress of this disease. And later on, we will get into details regarding this condition, the situation, and let's add a highlight that will be very relevant later. There's one key word. We'll put it in light orange. One key word here is, maybe you know it. Key word here is, here we go. Here it is. Overtaxed. Overtaxed. Yeah, that'll become very relevant later. Overtaxed. Oops. So, moving on. What other details are there? So now we can move on to figure 3. So there are two types of processing of pre-proglucagon. It can take place in the pancreas, or it can take place in the intestine. So not that many notes for this, it looks like, at least not for now. So when it comes to insulin, there is one polypeptide chain, and it gets made at the beta cells. And when it comes to glucagon, there are two polypeptide chains. I think I might have this backwards. Let's go back. Let's go back to the PDF. Okay, so glucagon is a single polypeptide chain. So glucagon, yeah, I have this backwards. So insulin has two polypeptide chains as noted here in this image. There you go. And there's the two peptide chains, 1 and 2. Good, good. That's a good correction. So moving on. Yeah, again, it's made at beta alpha cells. Glucagon is made at alpha cells of the pancreas. And here in parentheses, I also wrote plus intestine cells, and we won't specify for now. So here's a direct quote which came on page 10 of PDF number 3. Insulin and GCAG, or glucagon, they act in concert to maintain normal glycemia. Normal glycemia is a single term. So here is the figure itself. Pretty straightforward. I think it can be slightly more clear once it can be redrawn and things can line up better, and just color coded more extensively. So it doesn't seem too critical. Let's move on. So figure 4. This is about the control of insulin. This one gets kind of complicated. There's all these different types of events that happen in various places, and there's stuff that affects there's some steps or some events that affect other events. So let's get started. So first of all, and this is here because it becomes relevant later, but let's ignore this stuff over here for now. Let's ignore this. We will just be focusing on insulin release, all this stuff in the dark red letters. So there are several stimulants for releasing insulin. Here is a list of 6. Stimulant 1 is glucose, and then glucagon, leucine, ketone bodies, neural amplifiers, and incretins. I'll specify some details, some examples later. For now, let's move on to things which are suppressors of insulin. So insulin suppressors are glucose, but whenever there is a low concentration. So I guess here would be high concentration. So, but that wasn't specified in the notes, so let's just definitely specify here low concentration. And the second suppressor is suppressor number 2. The second suppressor is the P-sympathetic, P-symp, so parasympathetic input or stress. And we will cover details about the nervous system down there. So we'll get around to that. So moving on. So here are more deep details specifically about glucose, the first stimulant. So for now, we're not addressing a lot of the other stimulants. So there's 2 phases to the stimulation done by glucose. So during phase 1, the granules of insulin at the cell surface, those get released. And that ties back to this concept right here, step 7, where the there's insulin that's waiting near the surface of the pancreas cells, the pancreas beta cells. So that's what this is referencing. And then what happens during the second phase is, or the later phase, is that both old synthesized, here we go, sorry, that there's two types of insulin, insulin which was old synthesized and which is new synthesized. And that's the difference between phase 1 and 2. So moving on, there is a protein molecule named GLUT2, and this will be important to keep track of because there's GLUT2, but then there's also GLUT4. So what is the difference? Hopefully we'll address that in the near future. So anyway, GLUT2 is a transporter, and glucokinase is a glucose sensor. So that's in reference to, it's not labeled, but that's in reference to event 1 right here. So metabolism is just this gray blob because there's all those details from week 2 involved here, all of the glycolysis, citric acid cycle, etc., and all of the byproducts from that. So here, what is relevant is that during event 2, you experience some signals. And the first type of signal is an intake of calcium ions, which after you have that intake of calcium ions, event 3 happens and there is depolarization and some channels open, which it looks like maybe calcium ions go back out. I'd have to confirm with the notes, but later. Let's keep moving with the events. Definitely event number 4, which is seen here, is an event in which the mitochondria releases its stored calcium ions. So here you have the stored calcium ions in the figure. So now event 5 happens, and here's event 5, and as you can see, it's labeled immediate secretion. So that is the phase 1 of the release of insulin. And again, that has to do with this immediate release of insulin, which was ready to be exocytosed by means of granules on demand. So that's event 5, and then event 6, something that involves calmodulin. I don't have the details for now. I skipped those just to make this outline. Step 7, which happens here, also I don't have details for that. Just know that that PKC is involved, and PKC definitely becomes very relevant later. There's more notes here that will become relevant coming up. So anyway, event 8, which is seen here, event 8, it involves CAMP, and it also involves PKA. And later in the notes, not much became relevant related to that. So we'll know many details there. So now, here are some details in a similar fashion about another stimulant. So here we covered stimulant 1, which remember was glucose. But here we will be covering stimulant 6, which is incretins. So there's two types of incretins, at least in the context of this class. There's GIP, which stands for glucose-dependent insulinotropic peptide. And then there's GLP, which stands for glucagon-like peptide. Cool. And here's an important note. When you have a patient who has diabetes type 2, they lose the function of this first type of incretin. So they lose GIP function. That same patient keeps the function of this other incretin type, GLP-1. Moving on. Moving on. So figure 5. Yeah, figure 5 because that was 4. So yeah, figure 5. Here we address the nervous system. So here is a basic breakdown of, just the nervous system in general. You have your central nervous system, and you have your peripheral nervous system. We have these two to focus on, autonomic versus somatic. And for now, let's just focus on autonomic. And you have sympathetic and p-sympathetic, parasympathetic. So according to the notes, under the category of sympathetic, you need to take into consideration epinephrine. And remember, sympathetic, so let's go down here, parasympathetic. You can focus on the word para, P-A-R-A in Spanish. That means stop. So you can think of stopping to rest and digest. So parasympathetic is here, and up here is sympathetic. So this is the fight or flight. Hence why epinephrine does fit here. So two things to consider under sympathetic nervous system is the existence of alpha 2 adrenergic receptors and also beta 2 adrenergic receptors. So respectively, they down or decrease. When I write down, it just wanted to use a 4-letter word that's a synonym for decrease. So down. So CAMP goes down, which also means that insulin secretion goes down. And then in the case of the beta 2 adrenergic receptors, CAMP goes up. So also insulin secretion goes up. However, for some reason, I'll look this up in the notes in due time. For some reason, the net effect is that there is that insulin secretion goes down in general. It decreases. So now, in the context of the parasympathetic system, you must take into consideration acetylcholine, and there's another concept involved here called muscarinic agonist. Let's relabel this. We haven't used blue in a while, so let's use blue. Acetylcholine, something, muscarinic agonist. And by the way, here is the definition of agonist. So let's say you have substance, it is substance A. And the substance A, when it pairs up with a receptor, any receptor, fill in the blank, whatever kind of receptor, whenever there's an interaction between those two, there is the initiation of a physiological response. So, moving on, under the category of parasympathetic system and nervous system, the net effect is up, is that the insulin secretion goes up. And that makes sense because, remember, you're going to want to with the parasympathetic system, you're going to want to para, which means stop in Spanish, rest and digest. And that's when you'll need high or increased insulin secretion in order to be able to address the influx of glucose in your bloodstream now. So that happens through the activation of PKC, and again, that is related to, that's a throwback, that's a callback to PKC right here, this event right here, PKC. And yep, that's going to become very relevant later. PKC, that seems to be a major player in the context of insulin and also glucagon. So looks like that is all for there. And there was something mentioned, different types of diets, and there was an emphasis on this type of diet. I'll have to fill this out later to see how this diet varies and what are the different effects or the different types of things to take into consideration. So diet type 2, just happened to give that label. Diet type 2 is high in acid, in amino acids, and it's low on glucose. So what that means is that dietary glucose is not abundant, it is limited, so insulin secretion is low, and amino acid is absorbed by tissues in the liver. And I think that was a reference to the fact that these amino acids are going to undergo gluconeogenesis in order to make glucose and use that for fuel. So I'll come back to this as it becomes relevant. So here we start going into the new type, the next set of figures, which is from the new set of figures, which is from this PDF file, insulin action. Okay, is this a good time? Probably. Actually, no, we'll do that at the very end. There were all those side notes which we skipped for now, so we'll come back to those. So here's a reminder of something we addressed earlier. Insulin, function number 1, it promotes anabolism. And then function number 2, it inhibits catabolism. So here's some deeper details about that material. So the first, under the promotion of anabolic processes, insulin has 3 roles. Role number 1 is the synthesis and storage of glycogen, synthesis and storage of fats, and the synthesis and storage of triacylglycerides, of tags. For now, I don't have deep details about insulin metabolism, insulin's function related to the inhibition of catabolism. Moving on. So whenever insulin promotes these things, the first effect is that takes place at the adipose cells, and the uptake of glucose goes up. And now at a different location, at muscle cells, the same thing happens. The uptake of glucose goes up. And something else to consider in the context of muscle cells, the activity of this enzyme, glycogen synthase, that one also goes up. And this is a reference to something different. I fell back in addressing the, I fell back in studying. So there was no video for this, but here is the list of enzymes to which this is referencing. So here you have number 43, glycogen synthase, and that is related to let's go. Yeah, this is a good time to address it, otherwise it will make sense. So that's in reference to enzyme number 43 is right here. So here is this summary. This is from the step 1 book. So here's the summary of all the summary of the pathways, and they leave out a lot of stuff. So it's very simplified. So if you understand this, you're in good shape. I'm getting there. But anyway, 43, enzyme 43, again, was glycogen synthase. Where does that one get activated or get used? 43, here we go. During glycogenesis or glycogenolysis. Yep, they're in this orange space. So let's go back to the notes. Oh, I see. Okay. So let's go back to those other notes. So effect number 1 of insulin effect number 2 in the context of muscle cells is that the activity of enzyme number 43, which is right here, turning UDP glucose into glycogen, essentially synthesizing glycogen. Insulin is involved in making glycogen. So that's how things tie back into this diagram. Okay. So moving on then to the third effect of insulin promotion is that at the capillaries, you activate this other enzyme called lipoprotein lipase. And there weren't that many details for that, so we'll just be moving on. Let's make it orange. Lipoprotein lipase. So the effect number 4, the location of effect number 4 is at the liver. The formation of something called VLDL is increased. Glucokinase, I think it goes up. Yeah, I'm pretty sure the activity of this goes up. And also, similar to the muscle cells up here, there's also there's an increase in the activity of that same enzyme, which makes sense because in the liver is often associated with glycogen synthesis. So there are also effects to consider when it comes to insulin inhibition. The insulin inhibitory effects. So A is the inhibition of glycis, so glycogenesis. So okay, good. All this is tying well together. So whenever insulin is present, it will inhibit glycogenesis, which is what's seen here. You definitely don't, if you just use enzyme 43, you don't want that same substance, insulin, to also activate enzyme number 43, which would be glycogen phosphorylase, which from what we can see in this diagram, it's the reverse of what we just did, the breakdown of glycogen. So that's why glycogenesis is being inhibited. Good. So then you have effect B. It also inhibits gluconeogenesis. Yeah, that makes sense because you don't want to transition from glycogen to glucose. Good. And then the third effect is inhibition of lipolysis. We'll address that later. And also the inhibition of protein degradation. Cool. So that's all the details for that. This is all stuff to address before getting into figure 1. So here is just this overview, this tour of the insulin receptor. So each monomer consists of an alpha subunit and a beta subunit. So you have one monomer, and then the other one is essentially a mirror image. So at the alpha subunit, you experience something called negative cooperativity. Cooperativity. Yep. And then something noteworthy about the beta domain is that, yes, this is the domain, this is the portion, this is the subunit which is transmembrane. And outside the membrane, there are no noteworthy notes. Inside the membrane, however, you have the TK, the tyrosine kinase domain. That will become very important coming up. So anyway, so here you have figure 2 now. It's time to move on. So this is what happens. This is an explanation of the steps that happen in order to activate this receptor. So insulin, let's say it binds the alpha left subunit. So here you go on the left. So the alpha left, it possesses an IRTK right here. It's this circle, this oval. IRTK, remember, stands for insulin receptor tyrosine kinase, the ERTK. So what happens next is that the after this insulin binds, the ERTK left becomes yes active, and then that same ERTK left. Let's make this correction. The ERTK left, lots of stuff is happening with it. The ERTK left performs interchain autophosphorylation on the other ERTK, on the ERTK right. And then that means that the ERTK right becomes yes active, and then it performs just regular phosphorylation, not interchain autophosphorylation. That's why these letters are washed out. It just performs regular phosphorylation back onto ERTK left, and now there is full activation of the receptor. But moving forward, we'll just label it as act ERTK for activated ERTK. Cool. So we need to keep moving forward on this way. So figure 2, figure 4. Okay. Anyway, so now that you have an activated ERTK right here, the next step, number 1, is that at adipose tissues, you have the activation of the glucose transport system. And the same thing is happening at muscle cells, activation of glucose transport system. So after that, the next step is that at a variety of tissues, you have the activation of protein phosphatase, and protein phosphatase performs dephosphorylation. The dephosphorylation means that there is, yes, activation of enzymes which are related to fuel synthesis, and also the and then you also inactivate, so you have the not activation of enzymes related to fuel mobilization. So that makes sense. When you have insulin that became bound to the receptor, and then this downstream effect, this downstream cascade started happening, it makes sense that you will want to activate enzymes that make fuel, and then you are able to put it in storage, and you get rid of enzymes, or you take them out of commission so that they don't mobilize fuel, they don't begin to eat away at the storage. Nice, very nice. So keep moving on, still with the concept of act Urtica. The Urtica has substrates, and those substrates are called IRS, insulin receptor substrates. So the Urtica, it makes the IRS become yes activated, and the IRS acts as a docking protein. So here is dock partner. The dock partner number 1 is P85, and it looks like in the notes we didn't address any other partners. So, but we'll just leave this here as a as a fill in the blank in case we can expand on these notes. But anyway, just focusing on P85, we can begin to explore figure 4. So instead of being over here, figure 4 fits better right here. After we've examined this kind of downstream, this cascade of stuff that happens after insulin binds. So here is figure 4, and I didn't make too many notes on it because this one's actually pretty straightforward. Here you see that Arctica is indeed activated with all these phosphate groups. So that does activate on the IRS, and this active IRS goes on to interact with P85. And here you see that there is some involvement of these molecules, and then PDK is important because that activates AKT, and then AKT makes signals to Golgi to put these proteins on the membrane, and GLUT4 is yet another transporter. So here is the first instance where we see GLUT4 as opposed to GLUT2. Where did we see GLUT2 initially? Right there in figure 4. So same figure number. So there we saw GLUT2 and figure 4 over there, but also GLUT4 in figure 4. So in due time, we will build upon the relationship that exists there. So anyway, that's figure 4, and I think that's the end. Everything worthwhile noting. So then here's figure 5. It's pretty straightforward, and it's almost seems standalone, and it's very short, very small amount of material. Here is that material read verbatim. So here we go. So this subtopic is called regulation of DNA synthesis and cell growth. Insulin also regulates pathways in DNA synthesis and cell growth through the activation of RAS. The signaling pathway is initiated by activation of a protein named SHC. And this protein via GRB2 activates it activates RAS, which is a membrane bound G protein. And this should remind you of some of the concepts that we covered in cell biology over the summer. G proteins are involved in cell growth, so it doesn't look like we are required to dive into those details again, but definitely rings a lot of bells. So anyway, when RAS gets activated, it in turn activates a MAP kinase signaling pathway that leads to increased DNA synthesis and cell growth, which makes sense. You know, you have if you have glucose in the bloodstream, that made insulin get secreted at a higher rate. Well, so essentially, it's a way, eventually, it's an indirect way, but yet an effective way of signaling to the body that there is an abundance of food, so there can be a lot of cell growth that is allowed, that it's appropriate. Cool, all that's coming together. So here's where we begin to address on diabetes 1, type 1 versus type 2. So before doing that, we need to go back up to the top and address some of these side notes. So here in this side note is when it became appropriate, I think, to talk about all of these various omas. You have insulinoma, glucagonoma, etc., etc. So let's focus on insulinoma because it's related. It happens that it's related to pancreas beta cells. So Here are the notes related to that. It causes hypoglycemia, and there's something called the Whipple triad. I didn't go into details, but here is a short description of the triad. You have fasting hypoglycemia, and then you have hypoglycemia symptoms. And in regards to the hypoglycemia, the patient experiences resolution after IV glucose has been administered. And there's something about how 10% of the cases are related to something called MEN1, which stands for multiple endocrine neoplasia. So moving on to glucagonoma, it's something related to alpha cells. Okay, good enough. Let's move on. And then there's somatostatinoma, which is related to delta cells. And again, all this is within the pancreas. Cool. And then there's something called VIPoma. VIP stands for vasoactive intestinal peptide. So that's all the notes for now here. Whatever, we'll come back in more detail. So what else was a side note that we skipped? Here, very important. So we covered this event. Here's all these events listed in Figure 4 of this PDF file. Figure 4 of endocrine pancreas. So let's focus on event 7, which is this one. Protein kinase, and then that leads to insulin biosynthesis and processing. So event 7, the key player there is PKC. So what's special about PKC? Well, PKC promotes the production of something called VEGF, vascular endothelial growth factor. And VEGF, it performs neovascularization of diabetic retinopathy. Cool. Moving on. PKC, it also increases vasoconstrictors and decreases vasodilators. And PKC is related to accelerated atherosclerosis. And another related topic is diabetic microangiopathy. Under this topic, you can take into consideration 3 types of issues: nephropathy, retinopathy, and neuropathy. The notes focused on nephropathy, and they mentioned that renal failure is related to this concept. And within that other topic, that subtopic, is something called mesangial sclerosis. So I didn't go into details yet, but also keep in mind this other thing called nodular glomerulosclerosis. And obviously this is related to kidneys. And yeah, there you go, renal failure. All this is related. And then a specific disease name to start thinking about is Kimmel-Steele-Wilson disease. And we'll come back to that. Cool. So that's it for that side note. What other side notes were available? No, that's it. Cool. We covered everything that's. Oh, no, not yet. We addressed that. Yeah, it looks like we covered everything. So now, moving on to the comparison of diabetes 1 versus diabetes 2. Here's a table. What are some of the most, let's pick out 3 noteworthy things from here. I think this one's pretty important. The mechanism. Let's highlight that in yellow. The mechanism. So beta cells get destroyed under type 1. Beta cells get destroyed. Should make this bigger. That's a nice font. Maybe this. No. let's just make it bigger, whatever. 88. Nice. 88, and then here's 2. We'll make that bigger also. Cool. So the mechanism of type 1 diabetes is that beta cells get destroyed due to an autoimmune response. So M is for immunity, aut is for auto, immunity, autoimmunity. And when it comes to type 2 diabetes mellitus, the mechanism is variable and also unknown. A lot of mystery there. So the other thing that I find noteworthy is here, pathology. Well, pathology 1, because the 1 stands for first. So early pathology, you have insulinitis, and also you have reduced B cell mass, and beta cell mass. So, and here B cell doesn't stand for B cell for the immune response, it just stands for beta. So I just picked 50 as a way to symbolize the decrease in the B cell beta cell mass, and because later, you for sure it starts to get very severe once you do patho late, late pathology. Under diabetes type 1, you go from a reduced B cell mass to 0 B cell mass. Whew, that's troubling. As opposed to diabetes type 2, initially, the initial pathology, the first pathology stages, nothing's applicable. It's blank in the notes in the table. Let's confirm. Let's look at the table here. So this is under insulin action. So let's look at the table. Here we go. So first, here we go. Initial pathology, little or no pathology. There you go. Yeah. So essentially, I just chose to put in NA because I'm pretty sure in the notes it said that there are virtually zero warning signs. So so I was saying, let's focus also on late pathology. So here, where under type 1 you go down to 0% B cell mass. So your population of beta cells gets eradicated, and now you cannot produce insulin. That's a hallmark of type 1 diabetes, as opposed to late pathology of type 2 diabetes. The note said that you can either experience a lack of change in your B cell mass, it can remain normal, or if there is reduction, it's moderate. So here I just stuck with 100%, just to really highlight the difference between 100% beta cell mass and 0% beta cell mass. So something else notable under late pathology of type 1 diabetes, there's an increase in alpha cells and delta cells. So the increase, I just symbolized it with 120% and 120%. Here it's left blank because there were no notes about this, so I'm assuming let's just ignore things related to alpha cells and delta cells for type 2 diabetes. What is relevant is the following. There exists deposition, there you go, deposition of amyloid, and there exists fibrosis. And that's definitely relevant. Cool. So when it comes to genes, type 1 is, yes, linked to HLA genes, but when it comes to type 2, there is no linkage to those genes. Cool. So what else we got? Cool. We're almost done here. So here are the rest of the notes. So these notes were kind of monotonous. There were no figures, so this is the best I could get through them to understand this material. So there are three major topics left. Yeah, three major topics. Let's see. Let's label this also. You have just a topic named diabetes mellitus, and then you have this other topic which addresses three things: metabolic susceptibility, metabolic syndrome, and insulin resistance. And then you have this topic, diabetes mellitus, where the diabetes is considered mixed, and the focus of that section is the not adequate secretion of insulin. So under diabetic mellitus, you have this acronym, these initials H. It doesn't mean H in years. It stands for Advanced Glycation End Products. So the first type of H that was addressed, and there may be more, but in the notes there was only one addressed, and that's hemoglobin A1C. That's a big buzz term, and you hear that quite often. And finally, I got around with this class to learn about it. So hemoglobin A1C is a type of H, and there are effects which are mediated by H receptors. You have effect 1, which is the release of cytokines, which are pro-flam. That means flam is my shorthand notation for inflammation. So H receptors are associated with the release of cytokines, which are pro-inflammatory. Effect number 2 is the generation of ROS, reactive oxygen species. Effect number 3 is the increase activity of procoagulants. And then the effect number 4 is the proliferation of vascular epithelium. And then effect number 5 is the proliferation of ECM, extracellular matrix. So here's the other subtopic, pathogenesis of macrovascular, and also as well as, okay, the word and is necessary here. So pathogenesis of macrovascular and microvascular disease in patients with diabetes. I didn't write any notes here. Whatever, let's move on. The next major subtopic is metabolic susceptibility, metabolic syndrome, and insulin resistance. So there was a section that's not labeled, that doesn't have a subtopic name. However, this key term was encountered. Metabolic susceptibility. Cool. Moving on. Then the title of this subtopic is the role of fatty acids, and there's two concepts to address: inhibitory effects and circulating F acids. This portion of the notes got kind of complicated because there's all sorts of it seems like there are counterintuitive actions and effects, and this portion of the notes was addressing exacerbation of diabetes, exacerbation by obesity or by being overweight. So definitely there's more to be done to understand this portion of the notes. But for now, you know that fatty acids begin to be addressed, and also we begin to address the role of cytokines. So there's two types of cytokines that need to be taken into consideration in this topic. There are cytokines which are pro-inflammatory, and I wrote pro with two R's just to be able to have a four-letter word to counteract as a counterpart for anti-inflammatory. So anyway, there's three types of pro-inflammatory cytokines: TNA alpha, interleukin 6, and resistin. More notes are pending to come in relation to this. And then there's one type of anti-inflammatory cytokine, which was presented in the notes, and it's called adiponectin. Cool. Whew, we're almost done. So um, now here's this section which talked about mixed diabetes. So there were no subtopics in this at all. It just went straight into the notes, everything, and it made a difference. It differentiated two circumstances: the resistance whenever resistance was of low severity. Let's add some notes there. Of resistance of low severity and high severity. So whenever the severity is low, and I guess that means there's these are the early stages of diabetes mellitus. When resistance is low, yes, you are able to achieve balance. And the balance is between these two concepts: an increase in blood glucose, and then there's also an increase in insulin secretion. So the demand post the demand created by hyperglycemia is properly met by an availability of insulin to be secreted. So that's why this balance is achieved. However, later, when the resistance of insulin, the resistance to insulin becomes of high severity, you are not able to achieve that balance. You're not able to achieve that balance. The demand on the beta cells is too high. And remember, we pointed out an important term. Here it is, overtaxing. Let's go back up and find it also. It was here. So when it comes to diabetes mellitus 2, there you go, number 2, pancreatic beta cells can become overtaxed. Cool. There you go, everything. Well, this portion of the material is tying back to itself to over here, earlier in the notes. So anyway, let's go back down to the end of these notes here, and let's regain our focus on this key concept, overtaxing. There is something to take into consideration here called the incretin effect. I didn't take notes on that, but it does seem to be relevant. So the incretin effect. And what were the types of incretins? Oh, incretins. It's the incretin effect. Here we go. Yeah, so over here in the notes, we had gone into deep details about incretins, and the two types, or the two that were mentioned in the notes, were GIP and GLP-1. So incretin effects are relevant to the notes down here. So something about overtaxing the beta cells and the incretin effect. Cool. We'll come back to that. And then you have, there's another concept that needed to be addressed in regards to the high demand on beta cells, lipotoxicity. And there are some mechanisms proposed, and it seems like there's still debate in the scientific community about this, but what was provided in the notes was mechanism one, something about blunting due to obesity, and then something about alteration of mitochondria metabolism. That's what Sean is the shorthand for, mitochondria metabolism. And the third mechanism is induction of an uncoupling protein. And so definitely would pay it would be good to go back and review things about uncoupling proteins if you're rusty on that. I know I am for sure. So anyway, the three mechanisms in summary are using single words, blunting, alteration, and induction. And then one last thing to address. This was kind of standalone. I didn't understand it. I don't understand it yet. But there was this concept of amylin. So that's how all that fits in. Very good. So we'll pick this back up with further details after a little bit more study. Bye-bye.